It's a real pleasure to have the opportunity to be with you this evening to discuss the work that led to the birth of Dolly, the history of it, why did people start cloning, as well as what's the significance of it as we look into the future. One of the things which is always very important to make, one of the points it's important to make about a project like this is it is very much teamwork. And these are the group of people who are particularly concerned with the aspect of cloning itself. Keith Campbell has already been mentioned. He put a lot of the basic science into the project because of his background in cell cycle biology, which we'll get around to. Lorraine Young at the bottom left led a lot of research into understanding what was going wrong in cloning. Why did we quite often get unusually large offspring? Why was our success rate so low? And that was the area that Lorraine led. But over the years, this is the group of people who were actually involved in the project in different ways. And you'll notice that Angelica's name's in the left-hand column there. Quite a number of people on the list were employees of PPL Therapeutics rather than of the university. But there's a lot of different expertise there. The top and the middle there are two veterinary surgeons, one concerned with animal welfare, the other the pathologist who did the detailed examination of Dolly after she died. Molecular biologists, people who wished to use cloning to make precise genetic changes in animals. That was actually the reason why we started to clone, to be able to have cells in culture into which we could make precise genetic changes and then do nuclear transfer to produce an animal with that genetic change. That was the primary reason for the source of funding initially. This group here, bottom right, were all based in Aberdeen at that time. We had a project to investigate this large offspring problem going a number of years after the birth of Dolly, and that's the group that carried out that work. So, if you're going to get involved in a project like this, you need a substantial team, a lot of money, and good luck. And you'll see that some of the first thinking which led to the cloning procedures was actually carried out in the 19th century, well before people understood much of the developmental biology that underpins this work. They were asking questions, fundamental biology questions, which we still don't really have the answer to. There's a miracle goes on around us every day that a single cell of an embryo gives rise to all of the different tissues of an adult. And just think about it for a moment as to how complicated that mechanism must be. They didn't really know very much about chromosomes. They, of course, didn't know about DNA and genes at all. But they had some sort of idea of inheritance. After all, we all look somewhat like our parents. You can see inheritance in livestock and pets and so on. So they had a concept of inheritance, but very little of the molecular biology underpinning it. And we're thinking, what might it possibly be? And this was also leading to thoughts about whether or not you might be able to change cells, take cells of one type, do something and change them to something else which was more appropriate for some reason. And this was the gentleman who put forward an idea which is written out here. It's available in an English translation a little bit later than this, in the 1890s. It must follow that during segmentation and subsequent cell division, the nucleoplasm will enter upon definite and varied changes which must cause the differences in the cells that are produced. So they had that concept, that there is what we would regard as the nucleus, which governs many aspects of development and differentiation. The simplest hypothesis would be to suppose that at each division of the nucleus, its specific substance divides into two halves of unequal quality, so that the cell bodies would also be transformed. Now, this concept of unequal quality is a little bit sort of ambiguous, but in many discussions it's been taken to mean that if the cell that was being formed was going to produce muscle, it would lose the information that was necessary for, let's say, producing nerves, because it wasn't going to produce nerves. and the DNA would simply not be replicated. And it's that concept which led to the idea of cloning.